So hi everyone, welcome to our revision session. So I'm going to share my screen now and then I'll talk you through the PowerPoint. Okay, can you all see my PowerPoint?
Yeah.
Okay, fantastic. So the revision session starts now. So we're going to have an online quiz session and it's going to be opened on the 6th of May and will be open up until the 13th of May, midnight, and it closes. So you have that window to finish the quiz. Once you start a quiz, you have to finish it. So you can, oh, I'll come back tomorrow. So, and it doesn't matter when you want to do the quiz, you can do one on one day, another one on another day, depending on you, what you want to do. So there are 4 quizzes. The first one is hygiene, food hygiene test. It's quite, that one is quite easy and simple. The general food microbiology quiz is quite okay. The laboratory quiz is good and a bit, because I'm a bit worried because you've not done a hands-on lab. So I'll go through that particular, what to expect in that particular quiz a bit more in detail. But you did some observations, which is okay. Then the fourth one is microbial association with food. And we've done quite a lot in class in that area. But I'll give you a few tasters of that particular quiz as well. So moving on to the next thing. So I'm going to, what I'll do, the plan is I'll go over general microbiology and bring in the quizzes as they come along, yeah? So the first thing I want to talk to you about microorganisms, they are microscopic. So they're everywhere also, ubiquitous. So they're in the air, the soil, water, in and on plants and animals. Okay, so you can get microbes every part of our body. And say microscopic because we're not able to see it with the naked eye. So you need a microscope to be able to see it. The interesting thing about microorganisms, they're very, very, very tiny. So they are usually measured in a scale of 1/1,000,000th of a meter. And this is known as micrometer. Okay, so 10 to raise to power -6 micrometer. So you need to remember that. It's quite important that you remember that unit of measuring. Okay. So microbial world, when we talk about microbial world, what does that include and what does it mean? Generally, microorganisms are organisms that are too small to be seen with an unaided eye, and you usually require a microscope to see them. The thing about microorganisms is you have the good, the bad, and the ugly. So, the good one is that can be used for production of food. We'll have things like cheese, wine, and also for medications and stuff like that. But they also can cause diseases. I've covered quite a few foodborne microbial diseases in our lecture. And they're also responsible for food spoilage. And we've done a whole lecture on food spoilage as well. So they have the good, the bad, and the ugly aspect of it. So in terms of microorganisms, so it includes bacteria. And with the different types of bacteria and different classifications. So I expect you to remember some of the classification. They can be classified based on their gram staining. So what is gram negative? What is gram positive? And they can also be classified based on their morphology, so the shape, and also based on their oxygen requirements or temperature requirements. So I expect you to know those terms, and I'll go over it again with you today. So fungi, we have the yeast and the mold. They're normally, the spoilers. They cause food spoilage because, and they're able to spoil food that has low water activity. Mold is multicellular, while yeast is unicellular. Now, microscopic algae is another organism that is included in microorganisms. They will have the protozoans. They will have the viruses or viroids or prions. And these are non-living, and we classify them as non-living because they're not able to perform the 7 life processes. And we talked about this in lecture. So they are non-living. They're not able to reproduce by themselves. They need to get into a host hijack the host machinery, and then reproduce. Okay? Okay, if you do have any question, please send a message or chat, in the chat, and I'll address all the questions later or as we go along. So moving on then, what makes people sick from food then? What is it? So there are three classifications. So what can make you to be sick could be physical. So an object in the food that can cause injury, like glass, jewelry, bandage, staples, hair, or fingernails. So physical, or it could be biological. In terms of biological, germs that cannot be seen, like parasites, bacteria, viruses, we've talked about them, algae and stuff like that, those are biological. Or it could be chemical. These are poisonous substances like cleaning agents and pesticides that can be found in food. So these are the three classes of things that can cause problem, or if they're in food, they can cause make people sick or cause foodborne illness. Now, in terms of transmission, there are different ways microbes can be transmitted, okay, and get into our food so there are 5 key classifications of mode of transmission. So the first one, it could be waterborne transmission, either from drinking the water or swimming, or via fecal oral route, or fecal contamination of water from wastewater source or animal feedlots. So that is for waterborne transmission. Then we'll have the foodborne. So ingestion of the infectious agents. So if you eat a food that is contaminated with the microorganism, you can have foodborne infection. Poor sanitation is another route. Hygiene, So it's quite important that good hygienic practices are kept to in order to ensure that the food is safe. Another thing is insufficiently cooked fish and shellfish. And there's so many other reasons. We'll look at that later. This is just to give you an example of foodborne transmission. So waterborne transmission, foodborne transmission. The next one is airborne transmission. Okay? Inhalation of pathogens in aerosols or aerosols created at wastewater treatment plants, sludges or showers. So you have the fungal infections, viral infections, legionella diseases. And the next one is vector-borne transmission, animal and insect. So transmission by bite of an animal host, malaria, sleeping sickness, yellow fever. And the other mode of transmission, which is really not necessary for our food microbiology, is by sexual transmission, or exchange of bodily fluids. Those are the modes of microbial transmission, but that is not necessary for our particular lecture we're doing today. So now, what you need to remember, and it's very important that we remember that food which is free from contaminants will not cause illness, harm, or injury. So for food to cause problem, there has to be a contaminant in there. And do not forget, the foods that have pathogens might look, smell, and taste normal. Okay? If a food is contaminated by a spoiler, we can have the telltale sign of spoilage. Things like color change, of the smell. But that is with spoilers. Okay? So spoilers and how they can spoil food, and that will affect the organoleptic properties of the food. But in terms of the pathogens, you might see the food, it looks normal, tastes normal, and smells normal. So we need to remember that. It's very important. So if food gets into, if microorganisms, gets into the food, what happens then? You have foodborne illness, okay? And there are certain symptoms that are characteristics of foodborne illness. So you have things like abdominal pain, diarrhea, vomiting, nausea, fever, sometimes collapse, and dehydration. But this is not exclusive to, all the pathogens, so some could be some people experience some symptoms, others will experience other type. Now, if it was intoxication, so you have other neurological symptoms as well that will affect the nervous system. In the case of Clostridium perfringens, botulinum, Clostridium botulinum, sorry. Now, we're going to have one or two questions. So if, for example, we've all gone to restaurants and eaten, and then what happens is you can have an outbreak. So we're going to have a few questions, example questions on outbreak. What is an outbreak? So now, development of nausea and vomiting in 3 friends within 30 minutes of eating at a restaurant. Will you consider this as an outbreak, yes or no? Anyone?
Yes.
Yes, it's an outbreak because
Yes.
you have more than 2 or 3 people involved and the same, setting. Okay. Okay. The next question is diarrhea and abdominal pain due to Salmonella, Angona, an unusual type only seen once before in 24 persons in one state. Do you consider this as an outbreak? Yes or no?
Yeah.
Yes.
Yes, it's an outbreak. I mean, it's more than 2 or 3, so that's definitely an outbreak. The next question, a single case of botulism?
No.
No. Fantastic. So this is not an outbreak because you require 2 or more people exposed to the same common source for it to be an outbreak. Okay, so these are kind of simple questions, but it gets slightly interesting as you go along the quizzes. Now, the main reasons for foodborne illnesses, okay? One of the key ones is preparation of food too far in advance and then storing it at room temperature. Don't forget, most of the pathogens are mesophiles, and that means they can go in a normal body temperature, room temperature. So they'll multiply more in that temperature. So storing it at room temperature, they'll encourage these microbes to grow. The other thing is improper cooling, cross-contamination, storing hot food below 63 degrees centigrade because they're recommended to hot holding temperature. So if it's below, that could be a problem. They can start multiplying, especially the spore formers. Inadequate reheating of food, undercooking, low levels of hygiene, infected food handler, and consuming raw food. I mean, raw food, some of them needs to be cooked. So these are some of the main reasons for foodborne diseases. And you can see, some of the images here. So for example, the chopping board, you've used that to chop raw meat and maybe something to fry salads, vegetables. Of course, it could be cross-contamination. Raw meat is raw. It's got all the pathogens, that could contaminate it to be present. Okay? Unwashed hands, you've got here, and different other things. Flies, and unwashed knife, use it to cut chicken and use that to cut a cake as well. So these are some of the main reasons for foodborne outbreaks. Okay, one other thing we need to appreciate is that there are certain people in the population that could be susceptible, okay? And it's either because they're old or young, so pregnant or immunocompromised. So don't forget our acronym we used in Lendis was YOPI, yeah? So the young infants, the old, pregnant, and immunocompromised are the people that could be highly susceptible to a foodborne outbreak. So when we talk about foodborne, outbreaks, and an infection, so you can have foodborne infection and foodborne intoxication. What does it really mean? So in terms of foodborne infection, they are caused by eating the biological hazard along with the food. So if, for example, it's contaminated with the E. coli beef burger, and you consume the beef burger with E. coli in it, okay? So that's infection. So what happens? The E. coli gets into the gut, multiplies sufficiently, and starts to cause harm, okay? And there are different ways they can cause problems. The other way is foodborne intoxication. These are caused by consuming food that contains the toxic chemicals, and these chemical, or toxins have been secreted by the microorganism So you don't need the pathogen to be alive. So they're normally referred to as preformed toxin. Okay? So you need to also appreciate the fact that they have different symptoms that are peculiar to each one. For example, foodborne infection sometimes can have fever, characteristics of fever, and intoxication, the absence of fever. And the thing about it is foodborne infection, the onset time is longer than foodborne intoxication, which has short onset time. So you need to remember things like that as well. Okay? So intoxication usually results from consumption of food in which pathogens have grown and secreted enterotoxins. Enterotoxins. In most cases, intoxication is not dependent upon the growth and survival of the pathogen in the food. And many of the enterotoxins are potent, and small quantities are needed to cause illness, okay? So we need to remember that. And also we need to remember our toxin, what are the pathogens that are very common that produces toxins, preformed toxins. Preformed toxins. Now, do not forget, it's very important that you remember some pathogens, when you ingest the food with the pathogens in it, when they get into the gut, they can produce toxins. So that is different. That is one way they can cause harm. But in terms of intoxication, it's preformed. Okay? So that's the difference between the two. Preformed, so you don't need the pathogen to be alive, to be viable at all. Can someone give me an example of a pathogen that produces preformed toxins on food? Vicky, you're mute, so I can't hear you.
E. coli?
E. coli is infection, and it produces toxins when it gets into the gut. We're talking about intoxication now. Any other person? Okay, let me give you an example. Do you remember when we talked about the pathogen that caused the intoxication with the police force at Birmingham New Street Station area?
Staph aureus.
Staph aureus, yes. What is so special about Staph aureus? Can someone tell me what's so special?
It's very fast, the reaction.
Intoxication. So when you produce the toxins have shorter incubation period, yes? The thing about Staph aureus, you need to remember, we talked about it's everywhere. It's on our face, on our hands, on our body. So Staph aureus, when you have pimples or boils, it's got Staph aureus. Another thing about Staph aureus is considered an opportunistic pathogen, because what happens if you've cooked a food and you're not going to recook the food again, and an infected handler, for example, has got a wound, a cut on the finger, and they've not used plaster properly, and they touch the food, they can contaminate that food. And because you're not going to heat it again, Staph aureus can now start multiplying and producing preformed toxins, okay? The other thing also, because other food that is normally associated with Staph aureus is ham, because it's salted, okay, because of the salt in it. So Staph aureus is able to withstand, I think, 3.5% salt, okay? It's able to withstand salt. All right, do not forget that. It's important we remember that as well. Okay, so moving on then to microbes. So if a microbes get into the food, the next thing, we can do is, how can we tell is a particular microorganism that contaminated a particular food? So we can either examine it in the lab, and we've done a lab class, which you all observed, how when the food comes in, what do you do with the food? So, and also during food preparation, we need to know, is the food we prepared, or is this product we have produced free of contaminants? Because at the end of the day, you want to, sell food that is not injurious to, consumers. So what happens during food production, we need to know, is there any contamination of the food products? what is the food contaminated with? Because this will give us an idea if it's a food spoiler or a food pathogen. So if it's something like E. coli, Salmonella, Campylobacter, Clostridium botulinum, Perfringens, Bacillus cereus, that's a potential pathogen, you know? And also we need to know how severe is the contamination. Are the levels acceptable? And this will give us an indication of shelf life, you know, and, you know, when you're setting shelf life, and is there potential for a food poisoning outbreak? Or is the food, you know, going to spoil, you know, and hence change the organoleptic properties whilst it's out there in the market, and how long, you know? So, and also, so it gives us, so it helps assess the safety of the food for the consumer when we examine food. And also the microbial load and type are important in determining whether the food or food ingredients meet the acceptable standards. Okay? Specifications and guidance or guidelines. Does it meet it? And then you can say, okay, well, fine, it's safe to be released, or no, we're not going to release that food for public consumption. So now let's look at what happens when the food comes into the lab. So this shows the method of enumeration. Enumeration is to do with counting numbers. So when the food comes into the lab, you take a certain amount. For example, I've said here 25 grams of the food, okay? Or you can take 10 grams, as long as you remember what you have taken. Because when you're talking about the microbial load, you can refer it to per gram. So you need to remember, if it's 10 grams, you divide it by 10. If it's 25 grams, you divide it by 25 to give you per gram. So once you get the food, the first thing you do, you dilute it in with a enrichment broth or a diluent in a bag, okay? So then you homogenize it. The idea for homogenizing is so you can mix it up and dislodge if there's any microorganisms attached to the food particles, it dislodges. Then once you do that, you now do, it depends if you have pre-enriched it, you can incubate it for some time. But if you've not, then you can do a serial dilution. Serial dilution is very important because you do that because you want to get countable number of cells. Countable number of cells. Then after that, you now plate it on the plate, you know? So, and then you can now incubate it. Then you check the results, okay? And after checking the results, you know, by counting the number of cells, you can do other tests like observe it, look at it under the microscope by do gram staining. You can do some other tests like oxidase test and catalase. We did all this in the class. So in terms of preparing the plates, yeah, you know, you have to, because you're doing serial dilution, you need to prepare quite a lot of plates. Okay, then you plate them on, and you can see each colony here is representative of one a single cell. It multiplies in colony. So so that's what we call it colony forming units, because you can't really tell if it's just it originates from one, but it multiplies quite fast, so it forms a colony. Okay, so colony forming organisms. So it's not one colony, does not mean one cell, it means quite a lot of it in there. So now moving on then to serial dilution. So I just want you to imagine, okay, in terms of serial dilution, I've homogenized my cells, and this is my homogenate, this sample here, my homogenate. So I take 1 ml and I put it in a test tube. Then I mix it, take another one, put to the next test tube and mix it, okay? Then take another ml. So you keep going and going and going. The idea behind this is to have a diluted sample serially. So that will help us to count the number of cells we had in the original sample, okay? Now, you need to remember this and understand this. When you do the first dilution, because it's 1 in 9, that makes 1 in 10. 1 ml into 9, that's 10, yeah? So one other way of referring to that is 1 in 10 dilution. Then when you take 1 ml from the 1 in 10 to the next one, is 1 in 100. Then you take 1 mil to the next one is 1 in 1,000. Then 1 mil to the next one is 1 in 10,000. To the next, 1 in 100,000. To the next, 1 in a million. Now you need to understand and appreciate that, the more you dilute, the more chance of you getting a countable number of cells. Now, if we needed to recall how many we had in the original sample, so what we do is we now plate it on a plate, nutrient agar plates, or different types of plates, depending on the organism, because they all have different agars. You can, but the nutrient agar is a general purpose agar. So you can look at this one. So this is a 1 in 10 dilution, 1 in 10,000 dilution. Okay. So what we want to do, you take 0.1 mil. So this is 100 microliter. This is not up to 1 mil anymore. And when you plate it in that, whatever number of cells you have, you need to multiply that by 10, then multiply by the dilution factor, which is 10 to 4. So at the end of the day, you have 10 to 5. I'm going to repeat it again because it's a bit confusing. Now, once we've done our dilution and we decide to place it on the agar plate. So for example, if I was to take from the 10 to raised to power minus 5 dilution, that is a 1 in 100,000 dilution. I'm not going to take 1 mil to put on my plate. If it's a spread plate I'm doing, I take 1/10 of that a mil, 1/10 of a mil. So 0.1 mil, and then you plate it. Assuming I get 30 cells, or let me say I get 5 cells, but you're not allowed to count 5 cells because it's less than 30. So let's say assuming I get 30 cells, to know how many cells I had in my original E. coli sample here, what I need to do is 30 multiplied by 10, because don't forget it was only a tenth of a mil. So multiply by 10 to get it in a mil, to make it 1 mil. So that's 300. So 300 multiply by 10 to raise to power 5. So 300 multiplied by 100,000. And that will give you the number of E. coli cells I had in my original sample. Okay, is this confusing everyone? Yeah. Yes or no?
A little.
Okay, so we'll do the next one, then we'll go over it again. I know it can be confusing, so I need to take time and explain it, and I want to make sure you understand it. Okay, so let's go to the 1 in 1 million dilution. 1 in 1 million dilution. So when I done my serial dilution, 1 in 1 million dilution, so I take 1/10, so 0.1 mil, and I spread it on a Petri dish. Okay, so if I spread it on a Petri dish, and assuming I get 33 cells, okay, I need you all to have a go at calculating how many cells I have in my original sample. Let's have a go. So I have 33 cells there, and it's 1 million dilution. Okay? Okay. Ross, are you done?
No, I'm thoroughly confused.
Okay. So if I have 33 cells, I want to make sure if anyone understands it. Anyone done?
I've got an answer, but I don't think it's right.
No, okay. Could you tell me what you've done? Then I can see if it's correct. So you did 33 times what?
So I did 33 times 10 to
Yes.
the first bit. So I had 330, and then it's times by a million. 10 to the
Fantastic. That is it. That is exactly what you need to do. So it's 33 times 10 to get it to 1 mil, then multiply by the dilution factor at the top, which is 10 raised to power 6 times 1
But
million.
why does it say at the bottom 10 to the power of 7 you times it
Because when you
by?
do that, don't forget, because you're taking into consideration the first dilution. Does that make sense?
Yeah.
So you're taking that into consideration. So what that means is it's 10 times 10 raised to power 6. So 10 raised to power 7. Do you understand?
Okay.
Then I now multiply by my 33. Does that
Okay.
make sense?
Okay, yeah.
It's another way. It's just saying taking that one at the top and taking the other one is 10 raised to power 7. Then you now multiply by the number of cells you can count.
Okay.
We're
Okay.
going to have another go, okay? Because this is important. Why I'm emphasizing, because we have things like this coming out in the quiz. Okay? So
Okay.
yeah, anything I emphasize is because we're going to have that in the quiz. So let's go. We'll have another go. And if you're still confused, after the session, I can go over it with you and send you some more to work on, yeah?
Okay.
Okay, so let's look at another question here. So this is, I've tried to make it, so assuming this is our original sample, we took 1 mil from here to add to the first one. So this is the 1 in 10, the next one in 100, the next one in 1,000, and so on up to 1 in 1 million. Assuming we plated, so in this case now, I plated 1 mil. Don't forget, in the last one, I plated 1/10 of a mil. Okay? So you need to be careful. Check what did you plate. If it's 1/10, multiply by 10. If it's not 1/10, just multiply by the dilution factor. So in this other example I have, so I've put in 1 mil, so you don't need to multiply by 10. So looking at this now, you can see the first one is too much. You can't count. This is too much, so too numerous to count. We only count between 30 and 300. 30 and 300. Outside that, we do not count. 17, too few to count. 2, we cannot count. 0 does not mean there is no organism in there. It's just that we over-diluted it, yeah? So if I had done it with 1 mil, like this is just 1 mil I've added here, and I have 159, all I need to do is 159 times 10 raised to power 3. Okay? So 10 raised to power, so times 1,000. So 159 times 1,000, that will give us 1.5 times 10 raised to power 5. All you need to do, just count how many spaces before you put a point here. Yeah, before the decimal point. So 2. So you add 2 to the 3, and that's 5. But if I had used 0.1 mil on my plate, then what I need to do is multiply this again by 10. Does it make sense? Anyone?
Sort of.
Yeah.
Yeah, you break it down. So you simplify it down to sort of like 1 by the 10 power of 3. So you get 159 calories. And then to get to your correct figure, you're doing the times 10 by 3. But then what it means is then to actually simplify how many symbols you're going to use, you do the 1.59 by 10 to 5.
Yeah.
So you're breaking it down. So it's almost like the microbiological version of text speak, isn't it? You're sort of like simplifying it down, so you're not writing.
Yeah,
So you're not writing, 159. You're not writing down 15,000 or whatever. Are you?
Yes, exactly. Now, I'm going to ask a question now. Assuming I had in my 10 raised to power, that's in my 1 millionth dilution, which is the last one, assuming I had 48 colonies. 48 colonies. And in this case, I put 1 mil. 1 mil. Okay? Into the Petri dish. How many colonies will I have?
In which one? The last one?
The last one, in the 1 millionth.
$48 million.
Yeah, 48 million. Yes, 48 million. Okay, another question. Assuming I added 0.1 mil into my Petri dish, I didn't add 1 mil, but 0.1 mil, what will it be?
Would be negative.
No, it won't be negative. What did I tell you to do? If it's 0.1 mil,
480 million.
480 million, you multiply it by 10 to get it to 1 mil. 480. It's very important we appreciate this. Julia, we're going to do one more, and I need you to really pay attention. So on the 10 raised to power 5, okay? 10 raised to power minus 5 dilution, if I had 130 cells on my colony on the 10 raised to power 5, and I've used 1 mil for it, how many cells do I get? Anyone?
How many was it? 130?
Yeah, 130. Yeah.
Sorry, I completely zoned out. Where were we?
130, and
Yeah.
we've got that 130 in this dilution, 10 raised to power.
Okay.
Yeah, 10 raised to power minus 5. So all you need to do, I'm just going to say this again, we need to move on, is you had 130, you multiply by the dilution factor, which is 10 raised to power 5. So 130 times 10 raised to power 5. Then you can simplify it further, okay?
13 million.
Exactly. Yeah, I'll make it into a scientific way. And because it's 1 mil, if it's 0.1 mil, multiply that by 10.
130 million.
Exactly. Okay. So we're going to move on now. Now, after we've done this, and we need to observe see the type of organism we have, there's various tests we can do. And one of the things we can do, we can observe them under the microscope. We can do some gram staining. So I'm going to introduce the microscope to you. We had a look at some microscopes in the lab, but I want to tell you a little bit about the microscope. And don't forget, whenever I emphasize anything, is because I'm going to quizzes on that particular area. So looking at a microscope is really exciting, isn't it? So you can see the microscope has got an objective lens. So there's a lens there. And in some lens, cases, it could be times 10 magnification of it's called ocular, the eyepiece lens, ocular lens. Sometimes could be times 10, could be times 4, depending, okay? So there's a magnification going on there. Then you have other objective lenses as well. So these objective lenses are magnifications as well. So you can see when you look at them in the lab, you have times 4, times 10, times 40, and times 100, okay? So you have the arm where you use a lift in it. You have the focus wheel, and this moves this stage up and down, up and down. Then you have the base. You have the lamp that tries to throw more light into it. And then this is a slide we're going to observe, and this is a stage, okay? So, but what I need you to appreciate is what does the ocular lens do and the objective lens. So the next slide is going to talk about those lenses. So the ocular lens magnifies the specimen by 10. So that's the eyepiece, the one you used to look to see the organism. You also be looking through the ocular and objective lens simultaneously, okay? Because you need to use one of the lenses, you can see there. So multiplying the ocular magnification by the objective power gives us what is called the total magnification. Okay, so the red band, you can see the red band here. That is normally times 4 magnification. So if we're observing, so that'll be 10 times 4, that'll be 40. So I'm magnifying the object by 40 for the red band, okay? The yellow band is 10 magnification. So the ocular is 10, yellow band is 10. If we observe through the yellow, it's 100, okay? Now we have the blue band, which is by 40. So if I'm observing from the ocular lens to the blue band lens,
400.
what's my magnification, anyone?
400.
400, yeah? So then the other one is my black and white magnification. magnifies an object by 100 times. So when I look down, what is actual total magnification?
1,000.
1,000. Fantastic. Remember that, okay? It's important you remember that. Now, another thing I need to emphasize to you, this black and white one, sometimes we have to observe it through oil, okay? Immersion. So what you do, you put some oil on the slide, and then you put, you know, fine tune it. So this lens, the black and white one, touches the oil as well. And this oil immersion is a technique used to increase the resolving power, so you can see it clearly. And what happens, it uses the fact that oil has a high refractive index, okay? High refractive index. So look at the pictures at the bottom here. One is with the oil and the other is without the oil. So you can see with the oil, it's clearer. You can see it is, you know, giving you a better image compared without the oil. That looks more like out of, you know, you've not zoomed in properly. So that's the idea behind this oil immersion, okay? Try, you know, the refractive index of oil is high, and that gives us a better resolution. Okay, so when we look under the microscope, then we'll come to something very, very exciting. We'll come to different shapes, okay? So there are different shapes of microorganisms. So we'll have the coccus, we'll have the Coccobacillus, we'll have the Vibrio, we'll have the Bacillus, we'll have the Spirillum and the Spirochetes. Yeah, now don't forget, even with these classifications, we'll have variations in them. So like the coccus, for example, you have the ones that come Diplococcus, means two. You have the ones that come like a bunch of grapes. That is what Staphylococcus looks like, a bunch of grapes. You have the ones that like Streptococcus, like, you know, a string of beads, all, you know, and that is Streptococcus, yeah, string. So, and then you have the Bacillus, you can have the ones that come, two of it together, you know, Diplobacillus and stuff like that. So we can see the structures clearly once we have gram-stained them. And also we see them, and also we can tell if it's gram-positive or gram-negative. So basic characteristics, a classification of bacteria is based on the cell wall. So how do we classify gram-negative or gram-positive? It's because of the cell wall, okay? So there are two main groups, gram-positive and gram-negatives. Gram-staining is a differential staining technique that provides an easy differentiation of bacteria into two groups. Those that resist discoloration by 95% of ethanol are termed gram-positive, and those that do not resist it, and it can be discolored with ethanol, are called gram-negative, okay? Now, what is very important we remember, and the reason behind this is because the gram-positive bacteria have got a very thick peptidoglycan layer, so they're able to resist being discolored. Hence, they retain the purple color. You can see the purple color. While the gram-negatives, they have a very thin peptidoglycan layer, and the ethanol would discolor it easily. Okay. Okay, so we're going to have a look at some, and I want you to try and guess the shape. They're not very, very good, so the pictures normally, because they are, you know, from the microscope, but have a go guessing the shape and if it's gram-positive or gram-negative. Okay, so can you see it? It's not very clear, is it? Is it very clear? Okay. Number one. Who can tell me what is number one?
Negative.
It's gram-negative, but it's spiral. I don't know if you can see the shape.
No,
okay, so the bacillus is like, you can see the rod, those clear rods. Like number two is gram-positive bacillus. Yeah. Okay, you can see the rod shape, you know, clearly. I can see the gram, that other gram-negative. You see how, I don't know if you can see my, you can see how they're curved, yeah? Can you see that? Can you see my cursor? Yeah. So that is a spiral. So number three, who can tell me what number three is?
Okay.
Yes, gram-positive cocci. Fantastic. Okay, and number 4,
Bacillus.
yes, gram-negative bacillus, right? And number 5,
Cocky again.
yes. Yes. Fantastic. You're doing very well. That's really good. So you have places like this coming out just to make sure you appreciate the gram staining and also the shape of bacteria. So that's quite good. You got all the answers. So now, question number 1. You are looking at an E. coli cell with a microscope. The ocular lens has a magnification of 10 times, and the objective lens has a magnification of 40. What is the total magnification?
C.
C.
400.
400. Fantastic. You see, it's not that bad. Not that bad.
Even I could understand that one.
Oh.
That is so much.
Go to the next one.
40 times 10. Can you make sure that one's in the exam, please?
Okay, I'll try. I've already said the exam anyway, so I don't know how I can do that. Okay, question number 2. Anyone?
D.
Is it E? Spirilla.
Yeah.
Why do you think it's spirilla? Doesn't that sound like it's going to go like that, on and on?
Oh, yeah, yeah.
Streptobacillus. B.
No.
I think it's D.
What, D? Did anyone say D?
Yes. D.
D, yes. It's D.
Vibrio.
D. Vibro.
Vibro. Okay, so the next question.
Oh.
It's the outer membrane. B.
I've just told you this answer just now.
C.
Did you say B?
C.
Well, it could be actually C, yeah.
Let's see.
Oh, I just told you the answer.
It's D then, isn't it? The presence of the wax in my colleague.
D, C. Sorry, it's not D, it's C. It's
Yeah.
based on the thickness of the peptidoglycan layer. It's C.
Okay.
Russell, do you want to take a break?
No, I'm all right.
Okay.
Sorry.
Then
Lack of oxygen.
now, Russell, you're going to answer this question so I can make I need to be certain that you're paying attention. Okay, number 4.
A.
A. Yeah.
Hey, fantastic.
Yay!
So you're really paying attention.
We're on a winner.
Oh yeah, by the way. All right, then let's look at some of the microscope one because I'm a bit worried about the clarity. So this name the bacterial morphologies, shapes and arrangements seen here. So I've done the first one. Bacillus is number 3. Okay. So can someone have a go? Number 1, what is it?
Spirillum?
Yes, exactly. Fantastic. Wow, that is really good. Okay, number 3.
Cocky. Number 3 is Bacillus.
Bacillus.
Bacillus, okay, and it's gram-positive, okay? Number 4, can someone see number 4?
It looks the same. Bacillus.
Yes.
No, but what type of bacillus is it though?
Gram-positive.
Gram-positive, but what else? If you look at so when you have a
They're
question like
joined
that,
together.
yeah, exactly, diplobacillus.
Oh.
So you can see, if you look at the other ones, you can see there are two joints, yeah? So you'll be expected to look very closely if you're giving something like that. Okay, and what else we have? Number 5. Look at number 5 clearly. It looks different as well.
It looks like bacillus all joined together.
Exactly.
Mm.
So it's called streptobacillus. Like you have streptococcus, yeah? And then let's look at number 6.
Coccus.
Yeah, what type of coccus though? I don't know if you can see very clearly. they are normally is 2-2. So you have to try and 2-2. They kind of joined. So that is Diplococcus. No, 2-2 is Diplo. Yeah, Diplococcus. Okay. Let me know if you need a break. We can have like 5 minutes break or 2 minutes break if you do need it. If not, should I carry on?
Yeah.
Okay.
This is a kind of the lab quiz, yeah?
Yeah, this is lab quiz.
Okay.
But I've tried to bring in other things into it, but I've focused on like the magnification, the calculation, this looking at the morphologies, they're all part of the lab quiz.
Yeah.
Can you repeat what the last one was, please? Sorry, Helen.
The last one is,
6.
Can you see it on the board?
Yeah, but it's everybody's pictures are in the way.
Oh, really? Oh my God, I'm sure it's Russell's photo and who else is on the way? Can you see it now?
How come it's my fault?
You can shut the pictures up so you only see
I've
Helen.
done it.
No, that's
Yeah, I've done it now.
Okay, do you want me to shut it off?
No, that's right. I've shut that bit off now.
Okay, yeah, shut it off here. I want to
Thank
see
you.
everyone. Okay. So yeah. So I'm into the hygiene quiz now. What should we expect? I'll just do a little bit about each quiz is what to expect. Okay, the hygiene quiz. This quiz is designed to assess whether you have attained the necessary knowledge in food hygiene, which you all have. So it should be very easy. So there are a series of questions and each has multiple choice answers. Indicate your response and move on to the next one when completed. Don't forget, you're not allowed to go back. Okay? And one other thing you need to appreciate is this randomized quiz. And because it's randomized, it can come in any form. My question 1 can be your question 10. Do you understand? And because of that, please, when it says all of the above, and if all of the above is the first one, please accept, just, ignore that. Select all of below, if that makes sense. Because it's randomized, I forgot to change it to all. And I wrote all of the above when I wrote the quiz the first time. So if it says all of the above, what I mean is all the answers are applicable. Does that make sense?
Yeah.
Yeah.
I'm sorry about that. I'll see if I can modify it, but if I've not, and because it's randomized now, it can be all of your above could be number 1. Please don't get confused about it. I beg you. Just look at all of below. But I'll see if I can still modify because I've already set it up and sorted out. Okay, so let's have a look then. So why is personal hygiene important? It's quite important because bacteria live in and on the body and can enter into the food if we do not have a high standards of personal hygiene. So, like I told you earlier on, bacteria live on every part of our body. So our hands, hair, face, skin, clothing, and jewelry can be a source of bacteria, which can be transmitted into food. And this is known as cross-contamination. So you can contaminate the food by either touching your face, touching your body, and not washing your hands very well, or using the same chopping board for different foods. And there are different ways you can cross-contaminate. So this quiz will be trying to make sure you appreciate importance of personal hygiene. And in things like, okay, where do you store your food? The cooked one, where do you put it in the fridge, the top layer or the bottom? Do you understand? And, to just try to judge how good you are in terms of personal hygiene. So I've got a couple of questions that can help us here to appreciate the kind of questions will be there. So the first one is at work. The best way to dry your hands after washing is, number 1, using a warm air dryer. Number 2, give them a shake. Number 3, using a cotton towel. Number 4, using a paper towel. So what is the right answer here?
D.
D is the right answer. So you're good. See,
Yeah.
I knew it was going to be an easy peasy, lemon squeezy quiz. All right, now, food contaminated with food poisoning bacteria would smell, change color, look and taste normal, be slimy and bitter.
C.
What's the right answer?
D.
Sorry?
C.
C, fantastic, okay? Fantastic. Okay, the next one. What type of food hazard is it when you find a plaster in a food item?
B.
B.
Physical.
B,
Physical.
physical. Yeah, you see, you. See, it's very easy, honestly. You're going to be saying, Helen, thank you. Especially with this particular quiz, with this one, yes. But the lab one, you have to really try your best, please. That one is very easy, but just have to remember what we've talked about today. So general food microbiology quiz is like general food microbiology, what we've done. So I'll just go over one or two things with you and we'll have an example of the quiz. So remember what bacteria needs to grow. Remember factum? So they need a source of food, which is a nutrient, acid, so most of them grow at, what type of acid conditions they grow? In terms of acidity, what pH do they grow normally in?
Over 7. pH.
Exactly, yeah, about neutral pH. But some can grow at low acids, okay? Which bacteria will grow at low acid food?
Listeria.
Which one is known to grow in orange juice?
E. coli.
E. coli.
E. coli.
Now, let me tell you something. You know the quiz we did, who am I? Remember,
Yeah.
please go and bring it out because that goes through everything that is special about every microorganism. Because I took time to do, who do you think I am or what's so special about me? So if you remember that quiz, go and find it and recap on that. That will really help you.
What is that?
Huh?
What is that?
Who do you think I am? It's a quiz we did a while ago. We did it in class. So if you attended Helen's lecture.
Yeah, we did it. So yeah, I have to find. Yeah, we got the.
And if you can't find it, let me know. I can email it to you. And if you can't remember your answers, I'm quite happy for you to try answering them. Send it to me and I'll mark it. But we've done it in class anyway, so I expect you all to have a copy. But if you don't, it's very important you go through that because I'm not going to go through the pathogens today with you because you've got your crib sheet and you've got who do you think I am quiz that covers everything, that is special about every pathogen. Okay, the other thing they require is time. Then temperature has got to be right. So bacteria can be classified depending on the temperature of requirements. And I'm going to be asking you questions on this on the quiz. And oxygen requirements as well. And then moisture. So time and oxygen, I'll go through the classifications with you today. So temperature and bacteria, they have an amazing relationship, honestly. So in terms of killing the bacteria, so 100 degrees centigrade boiling water, bacteria will be destroyed. Cooking and reheating, will be destroyed. Minimum hot holding, they're not going to be able to grow. 37 degrees centigrade is an ideal temperature for bacteria to grow. Food storage, when you store it at this temperature or below, it will retard the growth of bacteria. It wouldn't grow, but they can grow, they wouldn't grow fast. They'll grow slowly because they're retarding the growth, okay? Refrigeration temperature, most of them, their growth will be retarded as well. But don't forget, some spoilers will still grow because if you leave your food, go on holiday, come back, they can get spoiled. But don't forget the pathogen that grows very well in refrigeration temperature. What's the name of the pathogen?
Listeria.
Listeria, yeah.
Ready to eat foods.
Yes, exactly. Then freezer temperature?
Minus 18.
Minus 18. Bacteria won't grow, but may not die. What you need to remember, okay, because people keep thinking refrigeration, they'll all die. Remember, when I store my microorganisms, I store them at very, very low temperature. And when I bring them back, I revive them again. So all the bacteria we use in a stock culture in a lab, even in all the lab experiments we do, we go and bring them back in those cold rooms, in the freezer, so they can, survive. They would not multiply, they would not grow. So what happens, we'll bring them out, we now thaw it, and we'll now subculture them, and they start growing again. Does that make sense? Because someone was telling me the other day, oh, Helen, even the viruses, if we freeze the food, you might kill them. I was like, how do you think we store bacteria in our labs? In the freezer. We'll prepare them in pure culture and we'll freeze them. Minus 18. When we need them, we'll bring them out in room temperature. They start growing. Does that make sense?
Mm-hmm.
So please do remember, they're not going to grow, they're not going to multiply, but that doesn't mean they'll die. Some may die, okay? some may die, but just remember the way that we keep our bacteria in the lab at the university, for a long time, a year, 2 years, 3 years in the freezer.
But parasites only might die.
Sorry?
Just maybe parasites on fish. Maybe.
Oh, yeah. Parasites and fish. I'm talking about bacteria, yes, because this is killing the bacteria. Definitely. They'll die because that's a control way of controlling them. But this case, I'm talking about bacteria. Okay, so temperature and microbial growth. So above the 63, temperatures in this range destroy most microorganisms, although lower temperatures take longer time. So the higher the temperature, the less time you need to kill it. Between 50 to 62, very slow bacterial growth, okay? Now, between 15 or 18, thereabout to 51 or 52, rapid growth of bacteria. Some may produce toxins, okay, in this range. Now, lower than that, many bacteria survive, some may grow. So that is between 15 to around about 4 or 5. 4 to 0, refrigeration temperature may allow slow growth of spoilage bacteria, but very few pathogens. So the one that we know that survives is Listeria. Lower, no significant growth. So this is not surviving, it's about growth, okay? Does that make sense?
Yeah.
All right, fantastic. Now, in terms of microorganisms and temperature, there are different classifications. So you have the psychotrophs, psychophiles, or sometimes referred to as psychotrophs, mesophiles, thermophiles, and hyperthermophiles. So remember, the ones that grow at body temperature, room temperature, they're considered as mesophiles, and it could be from temperature, like I told you earlier, if you remember, every microorganism has a minimum, optimum, and maximum temperature. So outside that is not very good, but within that they can grow. So outside a maximum, they wouldn't grow. Okay, and outside the minimum, they might not grow. So the optimum for mesophiles are like 37, 35, like body temperature. So psychotrophs, a pathogen we know very well, will be Listeria. Mesotroph, most of them fall under that. Okay, then thermotrophs, you have some bacteria that can withstand high temperature. So you're expected to appreciate and remember the classifications, okay, and have an idea of the range of temperature, okay? So question number 1. Now, I'll take this away because you can see. Okay, question number 1. Cultures of bacteria species were incubated on the shelf of a refrigerator, out on a laboratory bench top, on the shelf of a 37 degrees incubator, on the shelf of a 50 degrees centigrade incubator. After incubation, there was no growth at 37 degrees and 50. Slight growth on the bench top and abundant growth at the refrigeration. What term could be used for this species of bacteria?
Psycho.
So number D. So that's correct. Number D is the correct answer. Okay, question number 2. An organism with a temperature growth range between 45 degrees centigrade to 60 will be called a or an.
B.
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 Yeah? Fantastic. B. Number 3. All of the following could find a location on the body tissue for growth, except which one?
Facultad. Mesophyll.
Sorry?
D.
B.
D.
D. No, the mesophiles, they will grow on, they will grow. Mesophiles are body temperature, so you have most pathogens going there.
Is it cyclophile?
A.
It's A, exactly. Because our body temperature is not that low. That's like refrigeration temperature. Yeah? That's normally, body tissues. But then, exception is that Listeria. But Listeria can grow is able to survive and grow in low temperature. Okay? Now, I was expecting someone to ask me what E meant. What does E mean?
What does it mean?
Yeah.
I'll ask you.
High concentration of CO2. Now, one other thing we talked about that microorganisms require for growth, when we talked about factum, is oxygen. So bacteria need for oxygen. So you have aerobes. These are a variety of bacteria require oxygen to grow. And also most fungi and yeast are aerobic organisms. Then we have anaerobes. These are bacteria that grow in the absence of oxygen and are destroyed by oxygen, like Clostridium. So sometimes, you have obligate aerobes, obligate anaerobes. That means obligatory, must strictly. It requires that, yeah? Then we'll have facultative anaerobes. These are organisms that can grow in either the presence or absence of oxygen. So if we're to have a look at the, this in a test tubes, so you can see obligate aerobes will grow at the top, yeah, very close to where the oxygen is. Facultative anaerobes will grow everywhere, while obligate anaerobes will be found at the bottom of the nutrient broth or the broth. Aerotolerants will grow, you can see them everywhere because they just require very tiny oxygen. So they grow. The microaerophilic that require very, very, very tiny oxygen to grow. You see them growing in the middle. So it's quite exciting, isn't it?
Yeah, sure.
Very, very exciting. Oh no, do you know we're almost finishing? We have like 10 slides. I feel sad now. I'm sure Russell is happy. Okay.
I
Now,
was just thinking, so, well, do more, Helen. Do more. Let's see how many texts I get. Sure.
all right. Question number 1. Choose the correct answer to complete the incomplete statement below. Food spoilage is due to
A.
A, microorganisms and enzymes. Fantastic. Good. Question number 2. Preservation of food means
All of it. All of the above.
All the above.
when you preserve does not really retain the quality of food.
Isn't it C?
It's C, to prevent decomposition of food. That's the idea.
Mm.
That's the idea what we preserve, because we want to prevent the food from spoiling, yeah?
Yeah.
Question number 3.
B.
B.
Fantastic, we all got that. Now, the thing that is very, very interesting, and we've done this in class, is to do with microbial association with food. The relationship between microbial numbers and food spoilage is complex and depends on the number, type, and activity of the microorganisms present, the type of food, and also the intrinsic and extrinsic conditions. So it's quite a complex relationship. So what you're going to be doing, you're going to be giving some food, pictures of food, right? So you have the question that says, given the following food and their representative preparation and storage conditions, water activity, pH, select the microbe and pathogens most likely to prevail in that food. Okay? So microbial association of food with food is very, very important. That's the last quiz. You'll be given different scenarios and you're going to predict which food. But not to worry, some of those foods, we've done them in the class. But this requires you really thinking properly. and applying the knowledge. So what I've done, I've got a few samples, and if you look at the lecture, I gave you the pictures of food, and we talked about what pathogen would be in that particular food. So I'll just do like 3 of it here. I'll do the easy ones. So I'll leave you to the, when you get to the exam quiz, you see the easy peasy ones. So we'll have a vacuum packed hot smoked mackerel. The pH, so it's fresh fish with pH 6.6 to 6.8. They have smoked fish, water activity 0.8 and 0.9 of the water activity of the fish. So what pathogen is most likely going to contaminate this, if there is any?
Clostridium botulinum.
Exactly, Clostridium botulinum. Fantastic. You see, this one is easy. Imagine if I had like different types of salads with meat inside. You have to be thinking, okay, there's this meat, there's that as vegetable lettuce, there's this in it, and there's ham. But these, I'm showing you the easy peasy ones. Now, pasteurized fruit juice. Okay? The water activity is 0.87, pH is 3 to 4. What pathogen could be in this pasteurized?
E. coli.
No, when you hear the word pasteurized, what do you expect? You don't expect any pathogen to be present.
Should be nothing, shouldn't it?
Exactly. So you see the trick in these questions?
His pH, yeah, is under 0.94, and the pH is very low, so he had acidic. So
No, if it was unpasteurized, then you can say E. coli.
yeah.
But because it's pasteurized,
Treated.
does that make sense? So you have to be careful, just
Yeah.
check the words, yeah? Okay, the next one, rice salad in soya and oil dressing. Cooked rice, the pH is this, the water activity is that. What bacteria do you expect to grow?
Bacillus.
Bacillus cereus, yeah, because it's easy peasy. But if it was a complex one, you might have to, there might be more than 1 or 2 pathogens. So look at it carefully. And the last easy peasy one, dried packed pasta.
Pasta is Bacillus.
Yeah, suppose.
South.
Bacillus cereus, fantastic. So that's really good. Bacillus cereus is the answer, okay? So that's the kind of questions you have. You have the water activity given to you or the packaging. You can have a flapjack, you can have salads, you can have, all kinds of different types of food. If you remember, we did this activity in class, okay? And quite a lot of those activities we did in class, they are going to be coming out also in this particular quiz. So try and refer back to what we did, if you can find it. And then, I'm gonna miss you all. Remember your crib sheets.
Oh my God.
Go over who am I quiz. What is so unique about each pathogen? You need to remember that. But most importantly, keep safe. And finally, if we want to go out to eat, we need, can you see that? That's my joke. I'll just open to see if you can still laugh at my jokes. Yeah. Okay, so I'm gonna stop sharing now, and I'll stop the video. So the video, where's the video? Stop recording.